Okay, good afternoon, everyone. Is my audio clear?
Yes, sir.
Yes, okay, good. So I hope you are able to view my screen right now. And if we can try to solve this today, we will be able to, we will finish it off. That is today's class, basically. I hope you remember what we have done in the last class. We discussed about Southern blotting, right? Southern blotting to detect if a sequence is present or not. And we use that to find several genetic variations. Genetic variations could be, um, there could be insertions, point mutations, which include substitutions, insertion, deletions, and then large scale changes, insertions, deletions, inversions, duplications, and transpositions. We will also discuss a few of these in a matter of some time. But I hope you are able to recollect what we have discussed about Southern blotting. And yesterday I left you with trying to analyze this. So because you people also asked, I had one question that I usually use, and this is like trying to understand all blotting techniques at the same time. So we have three different blotting techniques, right? So before going there, I would want to tell something like this. So genetic variation, it could be it depends on whether it is going to have any consequences, right? When you say consequences, what does it mean? It's about the nature of the information. All the genome is genetic information. It is information. And actually, if you look at our own bodies, we are constructed because of our genetic information. In simple, or in probably the way which we don't realize, we are manifestation of the genetic information, or just simply information. We are what our genes are, which could, of course, be influenced by many other ways. But simplistically, if you are speaking, we are a physical manifestation of the genetic information that we have. And think of it like this: you are a person, and we are saying this is a embodiment of or physical manifestation of information, and you're trying to learn more about genetic information. That's nice, isn't it? Not many people do that. And it's nice. It's like information learning information. The point, what we are saying is, we are not just one person, we are so many people, and each have some differences, and that is what is the variation, the genetic variation. And if we have the genetic variation, then we want to study how the genetic variation is. Then we need to find out not just what the variation is, but the consequences of that variation. So there are two ways of how the consequences could change. That is, the variations could change based on the variations could change on the quality, at the level of quality, or at the level of quantity, right? When I say quality, say a protein A must have been produced. Maybe protein A is produced but with altered amino acid sequence at some point or some part. Or it could also be like the quantity. Maybe normally 10 molecules of A are produced, but maybe only 5 are produced, right? So these are the consequences. There can also be many genetic variations that may not have any consequences, right? For example, like the genetic code is degenerate, which means different codons code for the same amino acid. Could be. So genetic variations are what we are trying to study. Some of them have no consequences, some of them have consequences, and the consequences could be about the quality of the protein or the information that is being produced, or the quantity, how much of it is produced. There are more things to it if you look at it. The timing, when is it produced, that has to do with regulation. Say something should be produced at the level of when it is in blastocyst stage and not otherwise. You can also have, you can also say it as spatial, in the sense that some genes should be expressed only in the epithelium but not in the glands or say pancreatic beta cells, right? So any of these could change. If it is especially an eukaryotic one, these will change even more. These will become more important. That's why when we try to study genetic information at the lab for medical purposes, and it becomes, we need to look into all the details. And apart from that, not only, not just the person is not just the genetic information. There is also, it is also about epigenetics. I think we will have a discussion on that. If you all have studied stem cell biology or eukaryotic gene regulation, you might have heard about it. And we will read this. And I also, maybe in the next class, I will try to talk about it, because we will have a question on that in the coming CIA. And it is interesting in the perspective of interpretation and so on. Okay, I hope you understood what we are trying to do. In the previous class, we tried to see large scale, if we didn't bother about anything about the consequences of what is happening to the genetic the information itself. All we saw is physically, if there is a genome like this, what, how would it, how could we interpret if there is an insertion or a deletion? How could we find it out? That is what we've seen in the previous one using Southern blotting. Okay. So in the next class, what we will discuss, in this class, we are going to also focus on the consequences. So I hope, I hope we already know that from DNA or the genome, I'll just put it as DNA, that's easy. From DNA, that is, a gene is present, a gene gets transcribed, and it will give rise to RNA or mRNA, and the open reading frame could be there, and which upon translation would give rise to a protein. And we have discussed about Southern blotting in the previous class, which is to analyze if there is a particular sequence within a given DNA sample. For that, we would have used, for the technique, what we discussed is Southern blotting. The objective is whether gene X is present in a given DNA sample or not. If I want to find out if transcription of that particular gene has occurred or not, I would have used a technique called as Northern blot. So it is almost the same, except that we would have taken in Southern blotting, we would have taken sample DNA and test this whether sequence is present or not, gene X. Okay, we would have made a probe that would recognize or is complementary with gene X, and then through Southern blotting we would have known that. For Northern blotting is, we would have taken the sample RNA, which is the total RNA. We would have isolated RNA from cells, and then we would have checked for mRNA by using hybridization, like what we have used. We would have added a probe that would have been complementary to the mRNA for gene X, for example, if I want to say. And then we have another one that is called as Western blotting. that is used for, we would have taken the total protein of a cell, and then we would have done checked if protein X is produced or not. For protein X, we would not have used hybridization, like we would have used the probe here. In northern blotting and southern blotting is made up of nucleic acids, whereas here for detection we would have used antibodies. In all of them, they're all blotting techniques. You would have collected their respective samples. Okay, I will just put it as southern blotting, northern blotting, and western blotting, so that we can go through it and then eventually try to also do the problem that we want to discuss in the next slide. In southern blotting, you can think of it as sample is DNA. In northern blotting, the sample is RNA, and western blotting, it would have been the total protein, right? Then about the probe. Probe would have been nucleic acid based, and the same is the case here is nucleic acid. Probe here is antibody or monoclonal antibody. Okay, so by that, here the interaction is by nucleic acid base pairing, or say hybridization. The same is the case here too. And in this case, it is protein-protein interaction, that is antibody epitope interaction, or CDR epitope interaction. Okay, and in all the cases, we would have done blotting. In some cases, we would have used nitrocellulose membrane, in other cases, nylon membrane, to which the DNA would have been DNA or whatever we are trying to do. Check, we will be able to see it. In this case, in southern blotting, usually, it's not always the case. There are non-radioactive as well, but we use majority of the times radioactive elements for making a probe. The same is the case here too, radioactive elements. And in this case, usually it is enzyme conjugated ones. Okay, enzyme conjugated antibodies. Yes, there are primary antibodies and there are secondary antibodies. Okay, we will discuss these things again in detail. But here today, what I want is how can you relate these techniques and be able to think clearly when we have to analyze these techniques. Okay, so I hope you understood what I'm trying to do. If there is a mutation, say here is the promoter, what would happen if there was a mutation here? Would transcription be affected? Yes or no? Somebody can answer.
Transcription would happen, but the product
Transcription
would
would
be ahead.
happen, sir, regardless of what the mutation is. I mean, the protein which comes after the transcription and translation would be the one which has a difference, but the process would continue.
Yes, both are right. And transcription is not affected. The promoter region is intact, so transcription would occur. And does it mean that means the expression is unaffected? Is it right to say that statement?
No, sir.
We cannot say that because we don't know the nature of the change that occurred within the.
I mean,
Because,
expression would.
yes.
I mean, we can't say it that way, sir. But there is a difference in how it expresses. Maybe in terms of the sequence, or there would be change occurring, but unaffected expression is not the way to say. I would say there is a change, but
That's
not
right.
in terms of whether it stops or that it doesn't stop, but there is a change. Simple.
Yes, I would agree. But we don't have to really be certain about there will be a change, or we cannot be certain with whether there is no change. As I was talking to you, if it is a single nucleotide polymorphism and an amino acid codon has changed, but the codon still codes for the same amino acid. Okay, then the protein would be the same protein as it is. The transcription has not affected because assume that the promoter region is not affected, so the rate of transcription is same and the rate of translation could be same and the protein sequence has not changed. Okay, so to be certain whether there is a consequence or not is little difficult in the sense that we have to do more analysis of it. In the problem that we are going to solve today, I would also want you to think of all possibilities, not be sure, not don't be sure too fast. Okay, so here is a question we have and we need to get to the answer. How much time you take is how much, it's not about how much time you take, it's not about that, how much thought you have within that time. Everything intelligence is about, it's kind of for me, you can also think about how fast you can think in the yeah, the speed and accuracy or clarity of thinking is what is intelligence to me. That is how we get from a question to an answer. In many cases, especially in the research, there is a lot of uncertainty. You cannot determine the answer immediately, and there is, we will have to play with the many factors to think about. When we are thinking, we have so many things to consider, so many things to be considered as negligible and eliminate them, and so on. So I want you to think of all possibilities. Before going any further, I just want to also discuss a few things. Um, because we discussed about genetic variation, right? Insertion, deletions, single nucleotide polymorphisms, and so on. We have already actually done allele-specific PCR, where we can use primers to determine whether a mutation is present or not. Something like the genotyping. What we haven't discussed is about some other types in terms of at the level of protein, mutations describing at the level of protein. In that, we have nonsense mutations, we have missense, we have silent mutations, and we have, what is the other one called as? The frame shift mutations. Right? If you just excuse me 1 second, I will try to take a picture that I wanted to have. 1 second, be online. I would want to take a picture from that I made some time back to just illustrate these 4 things. Just stay online. So anyway, can you tell me what is meant by nonsense mutation? Anybody?
Whenever there is a stop codon before the actual stop codon, then that would be a nonsense mutation. A premature termination would happen.
That's right. That is a, yeah. Typically, we should have had the stop codon here. This is the start codon. Okay, if I'm putting it in, I'll put it in DNA, so that's easier. And the ATG is the start codon. You have to mug it up. TGA. What are the other stop codons? Quickly. Anybody?
TAG.
Okay. Keep saying. There is one more.
TAA.
That's right. So we have TAG and TAA. So I'll just put a comma, which would mean that each one of it is a codon, right? Codons are continuously continuous, and there are no interruptions. And in that case, if we have an insertion, if we have, say for example, here we had a GGA in the third codon, and if a transversion occurs, transversion, maybe I'll sorry, I'll put it as CGA I have. Okay, CGA, I have CGA, and it has changed to it has undergone transition. What would it become? I'm using, on purpose, I'm using technical terms so that you will probably get accustomed to those words. So what is transition?
Pyrimidine being substituted by a pyrimidine or purine substituted by a purine.
That's right. So what will the sequence change to?
TGA, sir.
Yeah, ATG will be changing to TGA. There is a codon here, then several codons, and then another TGA. Right? This is a stop codon. So transition is a purine pyrimidine changing to another pyrimidine, or a purine changing to another pyrimidine. In this case, it is pyrimidine changing to pyrimidine. So this is a stop codon. So that is a nonsense codon. Missense codon, or maybe I have copied the picture now. I will just paste it here and we will be able to see that. I hope you can see this picture, right?
Yes, sir.
So there are alleles, say for example, different alleles means there are different forms of the same gene, and there are mutations. First one is the X1 allele, X2, X3, and X4 alleles. Assume the top one is the normal one, which has about 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and TAA is the stop codon. So in nonsense mutation, the C here has undergone a, what did it undergo? It is a point mutation, it is a substitution, and what kind of mutation is it?
Transversion.
Transversion. That's right. A pyrimidine has become purine, adenine, and that is a stop codon, TAG. Okay, so that is a nonsense mutation. And what is a frame shift mutation? That is, here we have an insertion of G. If you compare with the TCG here, this is the original one. It would have been methionine, glutamine, serine, tyrosine, serine, and so on. Whereas here, because of an insertion, the reading frame has changed. So methionine, glutamine, serine, these two, until here it is fine, but from here on, it has changed. Glutamic acid, leucine, and stop codon. Okay, there is a shift in reading frame. So we call that as frame shift mutation. And then we have silent mutation. Where is the change? It's here, right? AGC codes for serine, and there is a change. It is a single nucleotide polymorphism. It is substitution, and it is transition, right? Because cytosine is substituted with thymine, and still the information has not changed. It is still coding for serine. So those are kind of silent mutations. Okay. So you have to remember, when there are insertions in frame shift mutations, and there are insertions in multiples of that are not multiples, insertions of a few bases that are not multiples of 3 within a open reading frame, the downstream of it would change the shift in the frame, reading frame. Okay, because ribosome always reads in triplets, and that is each of them is codon. Of course, there are variations to it when it comes to viruses and so on. There are sometimes the ribosome can move one step backwards or one step forward. These are called as frame shifting, ribosomal frame shifting. You can read up on it. Okay, so are we still in a situation to read this question? Let's try to read it, and I'm trying to read it along with you so that when a question is given in the CAA, usually it will be in this way. This is what the question will be. The problem will be with our ability to understand the whole data. What is in reading from, and if we don't understand any technical word, we might lose focus. Even if you're preparing for GATE or CSAR, especially in CSAR Part 3, where they test your research aptitude, you should be able to comprehend what the question is. And for that, it's easy if you can use your rough notebook to keep on writing what you are going through when you find a question like this. Okay, let's read this problem. And alpha interferon is encoded by a gene that does not contain any introns. So there are no introns. Normally, eukaryotic genes would have some introns. So the BAM-H1 restriction fragment containing the complete gene can be identified on a southern blot by hybridization to a specific interferon cDNA probe. So what was the probe? I hope during transcription, I have described to you in one time about the open reading frame. And the open reading frame, upon the gene transcription, we'll get mRNA. And I was telling you that this is plus strand. mRNA can be regarded as the plus strand, as the sense strand, and so on. And if we want to detect this mRNA, or if we want to detect whether the gene is present or not, we should have had some protein, some nucleic acid that is complementary to it, right? Only then it will be able to base pair. So what they have done in this particular experiment is that they have made cDNA, which is complementary DNA. There are steps which we will get to it at a later stage. So we'll also discuss about probe preparation later. So they have used reverse transcriptase to make a cDNA of this one. And then they have used it as probe for southern blotting experiment. Here, that is the southern blotting experiments. And we'll also discuss about how to radio label as well. Yes, hybridization using that probe under conditions of hybridization. And it will detect only interferon sequences. They're saying so to ensure or to say that there are no non-specific binding. Okay, this probe does not have any non-specific binding to any other part of the DNA except that it only binds specifically. That's the intended meaning of it. So to determine, the next part of it is to determine the cause of unknown immune deficiencies. Blood samples from the patients and unaffected people are screened for alpha interferon gene and its expression. So sample 1, I think, is the normal person, control. 2, 3, 4, 5 have deficiencies, some kind of deficiencies. Okay, the following drawing represents autoradiograms for southern and northern blots because they use radioactivity, and western blot probed by antibody, like I described a little earlier. Okay, person 1 and 2 are normal for immune capacity. So these two do not exhibit any kind of immuno deficiencies. They are fine. These two are fine. They are immunocompetent. 3, 4, 5 have immune deficiencies. Can we say what are those deficiencies? That's the question of it. Can we see one after one? And this is the only time I would be taking a regular class where I'm describing this thing. Otherwise, we will have to normally make another time to discuss these things because we are going slow for this question alone. So what does this band indicate? First question is that. What does the band there indicate? Yes?
That's alpha interferon in sample 1.
Yes, there is presence of alpha interferon. Because it's southern blotting, you can say the gene for alpha interferon is present. And do you expect any polymorphism? In terms of zygosity, not polymorphism, zygosity. I will call it as a gene I, okay? What gene, say interferon. So it could have had many alleles like I showed X1, X2 in the previous slide. like that there could be different alleles. So assume for the sake of it, allele 1, allele 2, allele 3 are there, or allele 4. You can have any number based on the mutations that are available. Yes, Parvati, you were saying something.
That it is homozygous for alpha interferon cells.
Yes. With so we will call this as the normal allele is allele I. So the first person, person 1, 1 second please, stay online. Yeah, sorry for the interruption. Okay, the person 1, we will take it as that person is homozygous, because if it is heterozygous, it would have been different, right? But this is not completely, we cannot say that it is true. Okay, what if there was a single nucleotide polymorphism? In that case, you would not be able to see two bands, right? You will only see one band, because the size of the gene would not have changed, or the fragment, if it is single nucleotide polymorphism. Keep that in mind. But for the sake of our understanding at this stage, with within the context, we will just go by this. So we will assume that because we say person 1 is normal, and for the context of the question, we will take this as, sorry, we will take this person as homozygous for allele 1. So I1, I put. So look at the person 2 from southern blotting. Yes, what do you think about it?
She's heterozygous, and one of the alleles is I1.
Yes, the person in sample 2 is certainly heterozygous. So 1 is corresponding to the band that we are calling the normal person. And it's also said in the question that these two people have no problems. So it could be that one allele, functional allele this person is having, the other one is something new, and that we will mark it as allele 2, I2. And what kind of, what do you expect in this case? What do you think? Why do you think it is like that? Band, formation of band there. How do you reason defining this one? Can you see it, or should I expand it? I hope you can see it.
Yes, sir, we can see it, sir.
Okay, good then. Yes, how do you reason a band formation here? Anybody, please?
So we could say it as since it's one from one allele and the other from the other, so it's like since you studied it before, it's one is acquired from the first one or the other is from the second. Since we could say it as heterozygous. That's one way of saying it.
Also, we can say that it has got some insertion, and therefore the band weight is higher, and that's why we are saying it is two bands.
Okay, I want to know why is the band up? If you have answered, sorry, I might have missed it again. It is heterozygous, that is clear. Why is the band up? What are the possibilities?
So the
Because
size
of
of
some
the fragment could
insertion.
be
Okay, what else is possible?
like she mentioned insertion. I would say the size of the fragment generated could be larger. I2, that is I2 has a larger fragment that the restriction
Why
site
would
has.
it have larger fragment?
Either due to loss of a restriction site, maybe.
That is right. That is also a possibility. It could be an insertion. It could be a loss of a restriction site. That is what I'm looking for. So either of those two are possible. Okay, just keep that in mind. I'm getting. So we are not concluding anything at this time. What we can say is that we have these. We have, it is heterozygous. Okay, person 2 is heterozygous, and this could be an insertion, or it could be a loss of a restriction site. Okay. And assume we are not looking at anything about Western blotting, Northern blotting, and Western blotting. We are only talking about Southern blotting. And then we can probably, if we think about all the possibilities right now, just looking at Southern blotting, and then we will be able to eliminate some of the things in the later slides after looking at Northern blotting and Western blotting. If we are starting to compare different blots and try to derive the most or the best possible information, reliable information, then we have to take this kind of approach. But upon practice, you don't have to really think as much. Some things will become very obvious eventually. Okay, so what about now? Can you tell about 3? Southern blot 3.
Homozygous for I2.
Homozygous for I2.
That is right. That is the most likely one. Okay, and for 4.
Homozygous for I1.
Yeah, but okay, for the moment could be, but the question says that these 3 people are affected. Although the bands are same, the people have 3, 4, 5 have deficiencies. That means it is not the same. The band looks same.
Maybe the deletions. Maybe there is.
Yeah, there could be some single nucleotide polymorphisms which cannot be detected.
Or either some substitutions or even some kind of deletion. Maybe
That's
not.
Right. Yes. Any kind of variation that does not affect the size. So you can take it as homozygous for I3 because they're not forming 2 bands. It could be. Okay. And 5. Well, 5 could be, as if we are discussing at this point like this, it could be I3, or maybe it is a different mutation, or it could be I3, I4, something like that. There are innumerable possibilities, right? Okay, till here we are. And for the sake of simplicity, what I'll do is I will consider this as a new mutation, and that the reasoning will follow in a while. So now we'll look at northern blotting. What is the difference between these two? First thing, what is the use of northern blotting? It is to detect the presence of an mRNA or RNA sequence within a given RNA sample. Okay, so it can be used for 2 purposes. First thing is if it is present or not. If you have a band, it is present. If not, it is not present. You can also, if you plan the experiments properly, you can also detect if it is more or less, if the expression is higher or lesser. Okay, that is another thing that you can predict. If you compare the samples 1 and 2 for the northern blot, what can you detect? What can you tell?
One can be told that there is no immune deficiency or something, like it's pretty much normal for immune capacity, that which is already given in the question. But
yes,
northern blot, it's evident enough to say that there is like there is no other kind of some mutations or any immune deficiency.
No, one is clear, right? The
One and
amount
two
of northern blot, it is clear that it should be normal. It is expressed in sufficient quantities. That's what we will take it as. What can we tell about 2?
2, since it's heterozygous, I think that's why maybe that amount of mRNA expressed might be a little less, like just by visually looking at it.
You're looking at this and compare. That means one of the allele is probably not having expression. The expression of this mRNA is less than that of wild type. We will say it as wild type 1. It is almost half, which would mean that 2 had 1 normal allele and 1 mutated allele, and from that mutation there is no transcription. So probably what you can say is I2 is having a mutation in which there is no that is affecting the transcription. Okay, I2 could be a mutation in which the transcription is inhibited. Okay, that is what you can think of it. I'll just move these there so that it will be I2 is, sorry, okay. So we know we can now try to have a good guess about I2 is the problem. There is no transcription from I2, and whatever mRNA is formed here is probably from the I1. Look at sample 3.
Since
If you
it's
look at sample
homozygous
3,
for
yes, it is homozygous for
I2, so
I2,
there's no transcription at all.
I2, and that is confirming that there is no transcription. If it is I2, no transcription, right?
Sir, but then I have a doubt.
Yes.
Sir, if that's the case, then we've previously in the previous slides we've just mentioned that transcription will still continue even though there is mutation.
No,
Only there
I
would be a
didn't
change.
say that. What I said is, I was mentioning, I think, very clearly, if mutation occurred here, okay, then it is not necessarily affecting the regulatory sequence, the promoter region. And in that case, transcription would occur. In the case that I am trying to describe now, the mutation may have occurred around this region.
Okay, so we would assume, or maybe it's almost clear that there is a mutation in the promoter region rather than in the open reading frame.
That's right.
If it's in the open reading frame, then we would have got a band.
It may have happened.
We
Yes.
would have had one having that there's a transcription which have occurred, but since the promoter might have been undergoing some kind of mutation or any way it is, I think that's why transcription has not occurred, because you need the promoter should be the
we cannot
specific
be
sequence.
sure about the nature of the mutation. What we can say, the consequence of the mutation is what we can see.
Like it's mutated. We could just say
Yeah,
that.
it is mutation, and the consequence is no transcription. So from I2, we do not have transcription. Okay? And that is why 3 is affected. The problem with 3 is mutation because of I2, homozygous for I2. And that's why there is no transcription for interferon, and that's why this person is affected. Look at 4 and 5. They are the interesting ones. 4 and 5, it's already decided that these 3, 4, 5 are having immune deficiencies. So what can you tell about 4? The transcription is normal. The effect is, there is no problem with transcription, right? Transcription is normal in both the cases, 3 and 4. So I will just put it as transcribed, and this one is also transcribed. Now we will look at the last bit of it. I should have written it differently, the order, but we will just go by it. So if you are looking at, now what we will look at is the Western blot. Western blot is for finding out if a particular protein is present in a given protein sample or not. So we would have isolated the total protein from cell or tissue, and then we would have performed Western blot using antibodies. And we will be able to then, because antibodies are highly specific to their respective epitopes, they would be, if they bind and we have a mechanism for finding out using radioactivity, or mostly it is enzyme based, then we will be able to find out the reason, whether if the sample has the protein of interest or not. So from Western blotting, one has high normal, okay, we'll just call it as normal wild type thing. So we don't have to really bother about sample 1. With sample 2, we understood that there is a mutation in transcription. And so there is only 50% of transcript present. And because of that, we don't have, we only have 50% of protein 2. So Western blotting, like Northern blotting, can be used for quality. I mean, whether it is present or not, whether a protein is present or not in a given sample. It can also be, if you planned it in a quantitative way, you can also say how much of the protein is present in a sample. While one has a certain amount of protein, the 2 has only 50% of it. Okay. And 3 if you look at, so did we discuss this? Yeah. So the mutation is I2 has a mutation in the transcription, and because of which you have less amount of mRNA, because of which we will have less amount of protein too. The quantity is affected, quantity of the protein produced, because quantity of mRNA is reduced, because there in I2 it has some mutation has prevented transcription from I2 allele. And this is also confirmed by this one, the way it is a context based one. So there is no mRNA, and therefore you will not have a protein. Okay. What is with, this is the problem. With 4, gene is normal, mRNA concentration is normal. And look at this. What is the difference, if you can identify? What is the difference here? And always we should compare.
I think the size of the protein varies in between 4 and 5, even though the mRNA have the same size or something. I think that's because of one, while doing translation, there is a change in the maybe in the codons or the way the
No, you can
translation.
talk in terms of the types of mutations we discussed today.
Yes, sir. Something related to that. I think while translation
What are the
occurs,
which type of mutation which is it possible?
frame shift. Maybe frame shift is possible because of I think while frame shift the codon also might change, and the I think the protein coding for that specific codon is I mean the amino acid, that is the coding for the specific codon might differ. So I think frame shift
Yes.
mutation is possible.
Yes. So all we need to know is that we have a protein of low molecular weight compared to that of the wild type. That's why it is running faster. Okay. Is it the same protein, almost the same protein as this or not? First question is that. Anybody wants to have a take on that?
Mostly not based on seeing the band. I think mostly no, but not sure as of it. Maybe like
Yes.
comparing the sequences we would know, but otherwise just looking at it, I would say no.
Okay.
My
Anybody
take
else?
would be
Yes.
different.
Yes.
I would say yes, because only then the corresponding antibody was able to bind with it, and therefore we are able to visualize it. But the function could be compromised.
Yeah, that is irrelevant. Whether functional or not is not we are bothered. We are about what we are bothered is whether is it the same related protein or not? Does it have any sequence similarity with the wild type protein or not? Any similarity at all?
It does,
So
sir.
yes, there is a protein, for example, and each of the proteins will fold into global structures, and these you can assume as epitopes which are recognized by the antibodies. Okay, antibodies, this region is somewhere the complementary determining region, and the region which the CDR interacts with, you call it as the epitope. The epitope is the part of the antigen that is recognized by that particular antibody. So a protein can have multiple epitopes. Okay, epitopes, each of them are different, and if you produced a monoclonal antibody for one epitope, it will only recognize that particular one. So I'm stretching this protein into linear, like something similar to that of a primary structure of amino acid, 1, 2, 3, and 4, just for sake of, it is not, this is not how it is, because protein is a 3-dimensional structure. For the sake of understanding, I'm trying to imply this. Say all the amino acids from here to here will form this epitope or domain, and all the amino acids here, assume that is how they are. Say if the, and you did not, I wanted to ask what all can cause truncation. First of all, is this a smaller protein than the wild type? Yes or no?
Yes, sir.
Yes, sir.
Yes. Okay. It is a smaller one. Then it must be a nonsense mutation. Right? Somewhere here it's possible that a mutation has occurred because of which this protein part is not produced. Assume like that. That is why you have almost same, but a smaller protein, and that's why it's running faster and yet be detected by the antibody, because probably in our case, in this case, the antibody might be, this is the antibody, and it is still present in here. If our antibody was something specific to this particular epitope, it would not have been recognized. So what we can say at this point about it is there is a nonsense mutation in I3, and that is it is a point mutation. It could be a single nucleotide polymorphism, a small one, because the gene size is not changing, mRNA size is not changing, intensity is not changing. It has not affected the gene, the transcription or and translation. What is affected is the nonsense mutation, because of which the protein is smaller. What can you tell about this one? For 5, yes. Do we have 5? Yes, I1. Yes. For 5th patient, what can we tell? So basically, one thing, yeah, because it became shorter, it is non-functional. Okay, that is why this person is having immune deficiency issues. What about person 5? Please hurry up. I think we are losing time.
The molecular weight is higher slightly when compared to the first one, the wild
Why
type
is it
protein.
higher? It is higher. That is very clear.
Maybe a similar thing what to the previous case we said that there is a frame shift mutation possibly, but in that case a codon might be there where the amino acid of a higher molecular weight could be present.
What do you mean amino acid of a higher molecular weight? Different amino acids have different molecular weights. That's why I'm asking. Did you mean that or anything else?
I mean, I'm trying to.
Yeah, that is all right. Don't worry about it. Just answer what you think.
I mean, not generally like for a specific amino acid. I'm not saying that, but maybe for the overall protein structure, that molecular weight could be different.
Yes.
I'm telling in terms of the overall structure. I meant to correct that. I'm not saying the particular amino acid molecular weight, but I'm saying for a whole protein molecular weight could differ because of the frame shift
Yes.
mutations. Maybe
Okay.
maybe that's the reason. Not
That's
very.
a possibility. Yeah, that's okay. We are all trying to answer. It's not necessarily the answers. We are trying to get to the best possible answer. Parvati, you were saying something?
So is it possible that the stop codon got mutated and some sort of a missense mutation wherein the stop codon actually coded for something because of some transition or transversion?
Yes, certainly that is one possibility. So the stop codon could have become something else, and then the ribosome would keep on translating until the next stop codon comes or the end of the mRNA comes. So the protein is same from here to here, and there is an additional part of the protein that is added. That is one possibility, yes. A stop codon becoming a sense codon. It is the reverse of nonsense codon. That is one possibility. What else is possible? Yes, anything else? Think of everything. You have to be wild when it comes to thinking. When we act, we have to be sensible. Right? Thinking, there is no problem with that. Nobody is affected. So think wild, and you can just get to an answer. What if? Yes, anybody is trying? I thought somebody.
So I thought it could be insertion, but the molecular weights of the in terms of northern blot and the southern blot, it was not reflected. So
Or
that's not
maybe
coming.
there could be an addition of it, something like insertion rather. Maybe an addition of an extra, something like that.
If one
Maybe.
base was added, okay, if one base was added here, then the TGA, sorry, the stop codon is no longer the stop codon, because the reading frame would have changed, isn't it? It would have gone until whenever the ribosome gets the stop codon. Is that right? Do you
Okay,
agree
so
with that? Yes. So there is also a possibility that a premature stop codon would have come across. But there is also a possibility on the other side. We are thinking about all the possibilities. But yes, I4, in that sense, I'll stop soon. Don't worry. I4 could be a what did I say? A stop codon becoming sense codon, or it could be a frame shift mutation that is happening towards the 3' side of the gene, because of which the stop codon is no longer a stop codon and the reading frame would have shifted. Because of that, the stop codon would have not become is no longer a stop codon. Then the ribosome would have translated until a stop codon comes by. Okay. Either way, these two proteins, although produced, are non-functional. If they were functional, these wouldn't have been patients. Right? Do you agree? Or you can also have other ways of thinking too, and you can share those with us.
I think I would agree with that, sir.
Yes.
Maybe.
Okay. Good. Yeah, you can think about it again and come back. You will have a question on this in the coming CAA. So it's important that everybody tries to understand. And if you have any questions, please get back, and we can always discuss about it. Okay.
Sure, sir.
Yes. Thank you. I think I took over your 6 minutes. Any questions then? No?
For now, no, sir.
Yes. Okay. Thank you very much. Thank you for the participation and coming to the class too. Have fun.
Thank you.
Yes. Bye-bye.
Thank you, sir.
See you. Bye-bye.